Hello there. So, in the last video, we discussed about the various stages of preclinical trials. If the drug is found to be promising in the preclinical trials, an application called IND or Investigational New Drug is filed with the United States Food and Drug Administration to get the permission for carrying out the clinical trials. Conventionally, there are 4 phases: human pharmacology and safety, therapeutic exploratory phase, therapeutic confirmation, finally, post-marketing surveillance. Recently, a new phase has been added to this trial, which will be revealed towards the end of this video, so try to stick with us till the end. Get on board. Phase 1 is human pharmacology and safety. It is an open-label test. That means both doctor and recipient are aware of what drug is being given. Performed on 20 to 100 healthy volunteers. Elderly and children are avoided, as this first phase is risky, and healthy individual is more tolerant to the adverse effect of a drug than any patient. But this method has a negative point too. As it is done on healthy individuals, the efficacy of the drug can't be determined. In some cases, when the drugs are too toxic for healthy recipients, this phase is done with the same number of patients. Like, in phase 1 trial of anti-cancer drugs. So, what's the purpose of this trial? It studies how toxic the drug is, and maximum tolerable dose or MTD. Also, the pharmacokinetic effects of the drug are observed. That is how it gets absorbed, how long it stays in body, how and in which form it's excreted, and all that. Phase 2 is therapeutic exploration. 200 to 500 patients considered. The study is mostly controlled. That is, patients are selected only on detailed review of their condition. It's single-blind, and that means the patients don't know that they are being tested with a new drug. Doctor knows. Extreme caution is maintained, and its primary aim is to determine the therapeutic efficacy as well as the dose range of the drug. Phase 3 is therapeutic confirmation. Here, 3,000 to 5,000 patients from various regions with different genetic makeup are considered. The phase is double-blinded, which means that both the doctors and the patients don't know. Aim is to confirm the therapeutic efficacy of the drug. After a successful phase 3 trial, NDA, that is, new drug application is submitted to the licensing authority like FDA, for marketing permission. Now, the 4th phase is post-marketing surveillance. As the drug is being marketed, the population under this phase is much higher. Through licensed medical practitioners, the data about the efficacy, acceptability by patients, and adverse effects of the drug is collected from the general patient population. The aim of this phase is to know about the effects occurring in the long-term usage of a drug. It's important to know that drug development is a long, complex and expensive activity. Suboptimal pharmacokinetics is stated to be a reason for failures during drug development. At the target organ, too low concentrations of drug for lesser time can lead to efficacy failures, while wrong concentrations reaching wrong targets for longer time may lead to toxicity. All this leads to wastage of money and resources. Thus, a new experimental approach has been developed, known as Phase 0 or microdosing study. A very low dose, 1/100th of the estimated human dose, or a maximum of 100 micrograms is given to each individual. This low dose doesn't produce any adverse effect, but the pharmacokinetic changes can be monitored with the help of sophisticated instruments. This Phase 0 studies have the potential to avoid costly animal pharmacokinetic studies and Phase I studies. That's why this special strategy is getting more popular day by day. With this we come to the end of this video on clinical trials. Let us know about any of your queries or feedback in the comment section. Hasta la vista.